like a 6 value, those are considered to be associated causally with the disease. So when she has done the total thousands and thousands of SNPs, she should have gotten at least loci which are linked to the type 2 diabetes which are already known. We know that the insulin receptors are associated, with type 2 diabetes. And we know that glycogen mobilization genes are associated. But unfortunately, she did not pick up anything of that kind. Okay? What is it telling us? It tells us that still technology has to be refined more. You're not going to get that easily. So you have some of the genes, only one gene which showed but which is not in that area somewhere here, but that is no good. That means the theory of getting this causal association still requires some kind of a correction or just improvement, and that is where the field stays at this point for genome-wide association analysis. But in fact, another group in England, actually they established what is it called a blood consortium. Okay? There is Netherlands, Belgium, and everywhere, all European countries joined together. Several labs, almost 200 laboratories, and pool their efforts. So what you can do is you can get patients from that place, this place, everywhere else, and analyze. In fact, they looked at thrombotic disease. Thrombosis is another polyfactorial. Okay? So they looked at those, and they found at least 81 genes which are causally associated with thrombosis. Okay? So because they found causally associated with thrombosis, how do they tell that these are associated with thrombosis? They could not. It's not easy. It's only a causal association. But if you want to show that these genes which are located in that area, let us say that you identified a polymorphism somewhere here. If you go to the human genome in that area, you may find 20 genes. Does it mean all 20 genes are associated with the disease? No. Only one of them may be associated with the disease. So how will you rule out those 20 genes to be there? So you have to come back to some other experimental model. There's no choice. So what, fortunately or unfortunately, for this group which has done genome-wide analysis, we have laid a lot of foundation in our lab for a zebrafish program, where we have definitely shown the path for the past 15 years that the zebrafish model could be extremely easily used for looking at the functions of the genes. So the European group found out that our model and method works very easily. So for these 80 genes that they've isolated from the humans, that they used our zebrafish model quickly to locate the genes of interest. So they used our model to knock down, and 6 or 8 genes were already identified that happened only last year or something like that. Okay? So we are fortunate because our model has been used and is very well recognized now in the entire world. And therefore, what I thought is it is appropriate for me to also tell you, even though that is not present in the textbook, I'm going to tell you a little bit about how in the zebrafish and how a gene can be cloned and how diseases can be studied in the zebrafish model. Okay? That is appropriate for you. So if at all a simple question comes from that, it is not present in the textbook, you have to follow this particular lecture very carefully. You're not going to get the information other than the information that you want to read my articles, you're going to get. So I think that is the end of this chapter at this point. So you could use this linkage analysis or polymorphic kind of a gene loci and do that. Either we can start today, or at least I will initiate the talk and then we'll continue in the next class before the exam. Okay? So that finishes 10 chapters for you. 10 chapters along with this small information that I'm going to give you would be on your test. Let me open this up. Now you want to stop?
Oh, I think she said something about stopping.
Oh, what? I didn't.
Oh,
I think you want to stop.
You want what?
I think he wants to stop.
Oh.
oh shoot. told you, we work on thrombosis disorders, okay? That is, if you get yourself punctured, a bleeding will occur. That means this particular tube contains a lot of information, like there are cells which are lining endothelium, there are cells which are flowing in the plasma. Beneath the endothelium, there is something else called subendothelial matrix and so on. So when you do puncture, you're going to clot and stop your blood flow out. That is called hemostasis. If any factor is missing, it doesn't matter. This is why it is polyfactorial we're talking about. Whether a factor on the red cell, a platelet, white cell, any cell doesn't matter. A factor in endothelium, a factor present in cells present in subendothelium, like fibroblasts are there. So you can have any factor that can be missing in this game, or factors which are pumping the blood, that is the heart. So all these factors can be detrimental and can cause you bleeding, because if the factors are missing. That is simple, that is outside. But if some factors are missing, it could also lead to the same clot that formed for outside to protect you from bleeding. It can also essentially make it clot inside. That is called thrombosis, that we talked about briefly before. If it happens in a vessel called vein, it is venous thrombosis. If it happens in a vessel called artery, that is arterial thrombosis. Look at the difference. This is loaded with not only platelets, those are those cells, but also some other cells, that is called red cells also come into play. That is why it's called red thrombus, and that is called white thrombus. Okay? It's all full of platelets or nothing but that. That is, arterial thrombus is white thrombus, venous thrombus is red thrombus. Okay? So I want to show you a small movie clip here, which you would understand what is thrombosis in this particular slide. This is the traffic in India. These are the veins and arteries. And look at here. There's going to be a small clot formation in these areas.
That looks so
These are small platelets here. These are on motorbikes and cycles. But after the clot forms, after some time, what happens is that this clot also dissolves, you see. And the dissolution of the clot is called fibrinolysis. Okay? So there is now no longer, so the clot dissolves. That means if you get a clot in the vessel of your heart, you call it myocardial infarction. But your body has the ability, even though clot is formed, your body has the ability to dissolve that clot. That is, if you keep quiet. It's not trivial. Once you're having a heart attack, you are going to be very nervous. You're going to have epinephrine. Lots of other things are happening in your body. It is not the clot that is killing you. It is the secondary reactions that you go through is killing you. So if you can keep quiet, it's just like a snake bite. When a snake bites you, if you're very completely quiet, it will not flow quickly into your bloodstream and destroy you. But is it possible to keep quiet? No, you will never do that. And that is where the difficulty comes in. Okay? In fact, when we were working on thrombosis in zebrafish, we found out that if you create a clot, I'll show you the clot creation in a minute. How much time is there for us?
6 minutes.
6 minutes? Good. We'll do something and then we'll see what we can do. So, if you create a clot, so what we're trying to do is, why should you study this clotting in a polygenic disorder? There's no way. People have already studied hemophilias. That means they have already isolated plasmatic factors. People have already studied platelet factors. A lot of platelet receptors are already known. But out of 25,000 genes, 300 laboratories in the world have been working on this field for 50 years. They could only come up with 150 factors so far. But if you take a look at Mendelian-McCusick calendar, McCusick volume, there are 3,000 genetic disorders. Okay, and even more, we don't know, it's not listed. So out of 25,000 genes, I would predict that there are 500 genes associated with thrombosis. Just an educated guess. How did I predict? There's no rationale. It is just because the reason what I used is 1/3 of the gene functions are known. 2/3 of the gene functions are not known in the genome. That means whatever this 150 is 1/3 representation. So 2/3 we still have to know. That is the simple blind logic that I used. Okay? That means 150 is known, at least another 300. So 300 plus 150, 450. That's what I said, 500 genes should be controlling the process of thrombosis. So that means there is some more room to discover these 300 factors newly. Okay? That is what the proposition that we did in the 1995. So at that time, how do you attack the problem? You cannot use this genetics like this, genome-wide associations. Even if I get it, I only causal relation. There is no genes are not going to be answered. That means we thought 15 years ago the best thing is to do functional genomics, not causal associations. There must be a model like Drosophila. If we can use such a model, we could probably get the gene functions very quickly documented. And the model I was looking for, I initially thought maybe Xenopus is a good model because Xenopus lays millions of eggs, maybe it's easy to do. But then I quickly realized it is easy to get a tadpole, but not easy to go from tadpole to frog. Not easy. It requires, fences around growing them. I had to go feed them non-vegetarian stuff, which I don't like to do. So it's pretty difficult to do the frog growing. Therefore, I was looking at the literature. Christiane Nusslein-Molhard from Germany popularized a model in 1990s, and that model is called zebrafish. And what she did was she was using this whole thing for a developmental program. The reason why she used it for a developmental program is because, I'm not going to go to the history of zebrafish because it actually comes from India. Okay, that is the first book that was published by a fellow called Buchanan in 1822, an account of fishes in the river Ganges and its branches from India. So this fish comes from India, not because that I am working on it, but because this fish has got several aspects. It is an excellent genetic model, and its embryos are absolutely transparent. See, look at that. Oops. Look at that. It is going through cell division after division. You can see all this gastrulation and blastula formation, and subsequently the cells will actually migrate, involute, and all sorts of, this is an epiboly, and it completely does that job very nicely, forming even somites. This is all happens within 19.5 hours. Extremely rapid. But if you take this whole yolk out completely, dissect it out, and keep this body and blow it up by going to the photographer, and take an early stage embryo of the human, put side by side, you cannot tell there is a fish or a human embryo. Absolutely the same. That means they're also having 25,000 genes. The entire development program is beautiful. Therefore, they thought we could study early developmental genetics and early developmental program very easily. But what I thought in 95 is that why is it that we cannot use this model for human disease application? Fortunately, we were the first group, as well as another group in Harvard, Len Zon's group, we were the only two who introduced this model to study human genetics. He was looking at red cell disorder, so he studied thalassemias. I was looking at thrombosis. Therefore, we were at the same time, we conceptualized that this could be used for disease processes.